Hello friends, in this video we are gonna learn the process of DNA replication, which is a very important topic. So first of all, as we already know that DNA replication is Greek was proposed by Watson and Crick as semi-conservative. Yes, it is a semi-conservative way in which the daughter or the new strand is comprised of the old parental strand and the new daughter strand, and hence it is a semi-conservative manner. But in this now we will learn how this replication take place. So first there is an origin site. Eukaryotes have more than one origin sites, but in prokaryotes we have only one origin site. What do you mean by origin site? Origin sites are actually the sites in the DNA segment. For example, this is the DNA segment. Here we have the bases aligned according to the complementary nucleotides. So we have the we have the origin sites. For example, as we know that in Escherichia coli there is double stranded circular DNA. In that we have only one origin site, means or there is only one point from which that DNA replication will origin. But in eukaryotes and in other higher organism we have longer DNA segments, hence we need more than one. Actually there are thousands of origin sites during S phase which are originated so that there could be replication of the DNA. So let's study how this happens. So first we require some of the enzymes. First there is opening of the DNA strand, which is done by helicase. So let's see, there is opening. Let's consider that this is the point where there is origin. So let me make this strand as, for simplification, as this, and there are the base pairs, and there is a formation of replication fork. This is done by the opening of the, actually there is breaking of the hydrogen bond between a DNA segment. The opening is done by helicase enzyme. And let me name this polarity 5 to 3, 3 to 5. So this is the strand, the polarity of the DNA segment. Actually we know that according to the Watson and Crick model, the DNA strands are anti-parallel. So this is done by helicase enzyme, but there is a formation of fork. This is stabilized by, there are certain proteins called as small strand binding protein. Small strand binding protein. So this actually stabilize the strand which are open. They don't allow them to again recoil. And since we are opening this, there is a formation of fork. Here there will be tension, since you can imagine it as a thread which is actually coiled like a DNA. When we open it from one end, the upper end becomes tight. So to release tension, there is an enzyme called topoisomerase, which there releases the tension which is there in the upper DNA strand. So now let's see how the replication take place. And now we will first need to know that the enzymes there are many enzymes. In prokaryotic we have three enzymes, that is DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. But in eukaryotes we have DNA polymerase alpha, beta, gamma, delta and epsilon. These are actually the Greek alphabets which we have taken as enzyme names, the types of enzyme in eukaryotes. So DNA polymerase actually have dual function, that is ligase and exonuclease. Ligasing actually means combining the DNA strands with the diester bonds, and which is actually the function of polymerase. And exonuclease is also the function of the DNA, which means removing of the DNA strands from the DNA segment. So here we need polymerization. We here need a complementary daughter strand here, which will be created. For this, the DNA polymerase cannot directly initiate this. For it needs a short RNA group. It needs a short RNA group which needs to be here so that it can add the energized deoxyadenosine triphosphate, which may again broke up to release the two phosphate groups and then combined to form phosphodiester bonds and again synthesize a new daughter strand. So for here there will be formation of RNA primer, RNA primer from RNA prime RNA RNA primase. This enzyme, this RNA primer is actually a sequence of base pairs which are here so that there is a 3 end free, since you know that phosphodiester bonds are only there from 3 to 5. So DNA polymerase actually needs, it cannot actually add on its own. So here we need actually a short DNA, a short nucleotide segment. So yes, now here there will be the DNA polymerase will act here, and this is the 5 end and this is 3 end, 3 OH end, and here the action will be in this direction, the polymerization will take place. But there is a problem in this strand here. If this stranding starts from here, then it will not be, since we know that if this is 3 to 5, the other strand will be from 3 to 5 here. But if we again start from here, if RNA primer comes here and then polymerization take place, then again it will be the same 5 to 3, but it has to be in the opposite direction. For this it starts here, there is RNA primer here and there's polymerization in this way, that is here it is 5 and here will be 3 will be coming here downside, and again there will be short segments which will be formed to continue this process. This will simply takes place in this direction, and the short segment will be known as the Okazaki fragments. These are the fragments actually, 3 to 5 short fragments. Here will be RNA primer, Okazaki fragments. So this is the template, DNA template strand. This is the leading or the continuous strand. Leading, continuous, since it is actually in a continuous manner, and here it is in fragment manner. This is lagging strand. Lagging strand or discontinuous, you can say it. So these are Okazaki fragments, and now there will be exonuclease action which will actually remove the primers from here and join them. Exonuclease will act on this. Exonuclease removes primers. DNA ligase joins the Okazaki fragments. So this is the actual whole scenario, as you can see. So let's actually write some of the enzymes which are there in prokaryotes. Enzymes, DNA polymerase. It is actually the DNA polymerase which actually do the exonuclease as well as ligasing activity. DNA polymerase 1, 2, 3. 3 is most efficient. It makes 2000 base pairs per second. In eukaryotes, enzymes, DNA polymerase alpha, beta, gamma, delta, epsilon, and this activity needs to be perfectly accurate, since there can be chances of inefficiency and hence there could be mutations. So these polymerases are actually fully efficient, and by this there is also an Errand detection method which actually keeps checking the DNA fragments. So yes, this is the case. Thank you.